welcome to Food Safety Info Friday.
I'm Mandy.
And I'm Teresa. Enjoy!
Hi everybody. So this week we are going to be discussing Listeria Right Now, which is the test we use at Riveridge to analyze your monthly swabbing for Listeria. So we're just kind of going to go into detail about how it works, the mechanism behind it, kind of take you back to Biology 101. And hopefully this will help you understand what I do in the lab and how your samples get processed. So first, we start out by collecting the swab, so collecting the sample. So hopefully these tubes look familiar. So first, what we do is we take this swab and we push it up against the sides of the tube, squeeze out all the broth in it, and then we dump out the broth. And then we swab the desired area, place it back in the tube, and then we take it to CIDR to be analyzed. So now we're back in the lab. So first thing we do is we add in a lysis buffer and we vortex, which simply means to mix it. So what does lyse mean? Well, it means to break open. So what's in the lysis buffer? Well, first off, there's enzymes. So hopefully this picture looks familiar to you. This was in the very first Food Safety Info Friday. And it's a small portion of the Listeria cell wall. So remember we have our really thick peptidoglycan layer. Remember we said this is super important for your chemicals to be at the right concentration so that they can penetrate this layer. And then we have our cell membrane. So what these enzymes will do in the lysis buffer is that they will come in and they will chew up this peptidoglycan layer. So think of enzymes as a little Pac-Man. They come along and they kind of eat away at this layer. Next, in the lysis buffer, there's salts and surfactants. So what will happen, first salts will have a charge on them, positive and negative, and what will happen is they will disrupt this cell membrane, which causes it to be disrupted and get all messed up. Surfactants, also known as detergents, will come to the cell wall, and we can see them in this picture. They're attacking the cell membrane. We see the cell membrane getting burst open, and all the contents that are inside of the cell will be released into the environment, into the tube that's in. And this is super important that all the cell contents get released, because we need access to some of these contents for the Listeria Right Now test to work. So surfactants, I thought this might be helpful since, we use detergents all the time, but we don't maybe understand what they do. So here we have detergent surfactant molecules coming into the cell membrane, kind of penetrating it. And we can see that down here, they are totally disrupting the membrane. There's holes in the membrane. And same thing's happening over here. Detergents coming in, they can encapsulate cell membrane proteins and cell membrane phospholipids and really break up that membrane so that the cell is not viable anymore, and then the cell contents get released into the environment. Next, we place these samples at 37 degrees Celsius for 10 minutes. Now, what is happening when we are doing this? Well, first, we're cleaning up around the nucleic material. Nucleic material is DNA and RNA, which we will get into later, but that's the basis for how Listeria Right Now works. So also, there's a protein called proteinase K, which this protein's job is to simply destroy other proteins. So how this works in our case is Listeria Right Now targets rRNA, which again we'll get into later. But what we need to know right now is that we start over here. So we're going to work backwards. We start with a protein, the ribosome. The ribosome is made into subunits, is composed of subunits, and those subunits are composed of rRNA and proteins. So what proteinase K will do is it will come in and it will disrupt this whole protein to get down to its components. All right, the next step is to place the samples at 80 degrees Celsius for 28 minutes. And this step is important because we want dirt and other particles to settle out. So when we have a really dirty sample, we may leave the sample tubes in at 80 degrees for a longer period of time. Next, what happens in this step is also a deactivation of cellular enzymes. So what does that mean? Well, we can see over here, we have a functional enzyme, and a functional enzyme has an active site. This active site is kind of like the factory portion of this enzyme. So for example, we have apples as our substrate. The apples will enter into this active site, and then the active site, when the enzyme is functional, will make an apple pie. So push out a different product. However, when we heat up the tubes to 80 degrees Celsius, which is a really high temperature, a thing called protein denaturization will happen, which simply means that this used to be functional protein is all unraveled over here. And we can see that the active site is now kind of spread out, so it's not really functional anymore. So now we have our substrate, apples. Well, they can't bind into that active site factory to get made into apple pie. So this protein is no longer functional. This is important for us because we don't want any other products interfering with our analysis of Listeria. Next, we're going to heat the reaction tubes for 4 minutes at 56 degrees Celsius. So let me explain the difference between our sample tubes and our reaction tubes. Here are the sample tubes. These have our lysis buffer in them, and these are the tubes that have the dirt that was settled out and our denatured proteins in them. These are our reaction tubes. These are what contain the materials for the analysis of the Listeria RNA. So why do we do this? Simply, we want to get these reaction tubes to the correct temperature. So just like how we operate best at 98.6 degrees, these tubes operate best at 56 degrees Celsius. So all enzymes have an optimal temperature, and these tubes contain enzymes that need to be at 56 degrees. So let's pause and let's recap what has happened so far. So first, our cell was busted open with the lysis buffer. Our rRNA was made readily accessible by proteinase K. Then we degrade all their cellular components so that no interference is going to happen with our analysis. So now after all these steps, we're finally ready to run the tubes or run the samples. Okay, so first we're going to transfer 50 microliters of sample into heated reaction tubes. And that was seen in the previous slide. We're going to cap the tubes and we're going to vortex them for 2 seconds. So you can see in the video, I am quickly vortexing them. And then we are going to flick the tubes just to get out any bubbles that are in the samples so that these bubbles don't interfere with the test. Now this process of transferring and flicking the tubes and recapping them and getting them in the machine all needs to be done in less than 1 minute. So you can see me placing them in there, closing the lid, and now we immediately start the test. And at this point, everything that I physically need to do to the samples is done. Now the machine takes over. So let's pause from the method and let's go into some cell biology 101, because this is important for understanding how Listeria right now works. So first we have DNA, also known as deoxyribonucleic acid. This material is the basis for all living organisms. This is what makes you, and me. So DNA has 4 nucleotides. We have A, T, C, and G: adenine, thymine, cytosine, and guanine. So what you need to know, A goes with T and C goes with G, always. This means that A is complementary to T and C is complementary to G and vice versa. The orders of these base pairs is the basis for what proteins are produced. So let me explain to you this way. In the alphabet, you have individual letters, and those individual letters make up a word. Same thing kind of goes for DNA. We have our individual nucleotide bases. The order of these bases will make up a word, and we use words to convey messages to each other. So the order of these nucleotides will convey a message to the cell of what protein needs to be made. From DNA, we make RNA. RNA stands for ribonucleic acid. So here's the difference. We can see we have DNA and we have RNA. DNA has thymine, like we said, but RNA has uracil. And this is the cell's way of knowing the difference between DNA and RNA. And there's 3 different types of RNA. First, there's messenger RNA, and this is used kind of as a translator between DNA and protein. Next, we have transfer RNA. So transfer RNA is this kind of weird T-looking structure over here. And simply the purpose of this tRNA is to bring in amino acids, which amino acids make up proteins. It helps bring these amino acids together to make up a protein. Finally, we have ribosomal RNA, rRNA, like we already talked about. So again, we have this picture, and rRNA, like we said, makes up the ribosome. The ribosome is responsible for putting the proteins together. So what will happen, mRNA, tRNA, and this rRNA will all come together in the ribosome to make a protein. And ribosomal RNA is the basis for Listeria Right Now and what it is measuring. So now we have some background into kind of the biology of Listeria Right Now. Let's talk about the answer machine, which is the machine that we place these samples in, and then this machine will give us a positive, negative, or invalid result. So answer stands for amplified nucleic single temperature reaction. So what this means is the temperature remains constant at 56 degrees Celsius for the entire time of the reaction. So our target for Listeria Right Now is an rRNA gene. And there's about 1,000 to 10,000 copies of this per cell in Listeria. So it's present in all Listeria species. So it's present in Listeria monocytogenes and Listeria, whatever. It's present in all of them. So here's how this works. First, we're going to start up here. Remember, we have our rRNA, or ribosomal RNA, and it's single-stranded. What will happen is a process, and a protein called reverse transcriptase will come along, and it will make this RNA into cDNA. So what do I mean by this? So rRNA, I said, was like a translator. So let's say DNA is someone who speaks English, RNA is someone who is bilingual, and then a protein is someone who speaks Spanish. rRNA can act as a translator between the two. So we have our rRNA, and a complementary strand of DNA is made. So remember we talked about complementary nucleotide bases? So the same thing will happen. We can see over here, we had some A's on the end, and our cDNA matched it with T's. From this, these strands will be separated from each other, and then this single-stranded DNA will be made into 2 strands. So now it is double-stranded DNA. Next, what will happen in the reaction, we have nicking enzymes, which are like scissors. So in the green, we have our double-stranded DNA, and then we have a enzyme called a DNA polymerase will come in here and simply make more nucleotides and add them. A nicking enzyme will come in and clip this portion of the DNA, and then this little segment will be released into the environment. And this process of the DNA polymerase coming in, making strand, nicking enzyme coming in, clipping it off will keep happening during this process. So now we have our segments that were clipped off, as in the previous slide, which are right here. These are released into the reaction tube environment. Also in the reaction tube are probes, which is basically an identifier. So what will happen is these probes will come and find their matching or complementary sequence of the released Listeria fragments. Once they attach, it will fluoresce. And this fluorescence is the basis for how Listeria Right Now is measured. So now we finally get to our analysis. So these are some graphs that some of our packers have had. And so we can see we have a positive graph right here. The green line is the control. The orange is the sample line. So we can see here the control line rows and the sample line rows. So this indicates a positive result. Over here we have a negative result. We can see that the control line rose, which means that the test worked, but our sample line did not rise at all, which gave us a negative result. And now we have the infamous invalid. So here we can see neither line rose. This control line did not rise, which indicates the test did not work. So why is the internal control so important? Well, if the control line does not rise like it should, then some sort of inhibition has occurred. So how the internal control works is that there is a dye in the reaction tubes which will bind to that double-stranded DNA that we talked about, and it will give off a different signal than what the probe will. So if this signal cannot be read, then the test will not work. And here are some reasons why. We know dirt, wax, and sanitizer. So dirt and wax will prevent light from being emitted, which makes sense. If the control cannot be detected, then it's certainly not going to detect the probe that's looking for our RNA fragments. So again, we have kind of a dirty sample. This one was invalid. Wax, definitely no light is getting through there. But we have these other ones down here that, they look fine. Light should get through there, but one of these may be invalid because of sanitizer. And here's the reason why. Sanitizers cause a salt imbalance. And like we said earlier in the slideshow, salts can affect the permeability of the Listeria membrane. So these salts will, if you have excess sanitizer, will cause an issue with Listeria Right Now. And this is why. DNA polymerase, which is needed for the replication of DNA in the answer reader, needs magnesium. It needs it to function. However, if magnesium is bound up with other salts that are in your sanitizer, it cannot properly replicate the Listeria DNA. So this is why we can get an invalid. Therefore, if DNA can't be replicated, then the dye, the internal control, cannot bind to the DNA to cause light emittance. So I hope this helped. It was very sciencey, but I think it was important that we all at least have somewhat of a general understanding of how our Listeria Right Now machine works. So if you have any questions or would like me to explain more of it, then I would be more than happy to do so. But in our upcoming videos, we are going to examine the differences in the analysis methods. So We have our basis for Listeria Right Now, so now we can examine, what is the culture method really about, and how do these methods really compare, or do they even compare at all? We're going to talk about steps to improve your environmental program. Looking at, do you have a site, a Listeria site picking list? And then finally, we're going to talk about ATP testing and trending. So thank you for watching. Hope this helped, and have an awesome Friday.